Hello students, in this video I am going to tell you about enzymes. What are enzymes? Enzymes are biological catalysts, and because they quicken most of the chemical reactions occurring in the organisms, they have been designated as the manifestations of nature's impatience. Now the definition. Enzymes are biological catalysts. They are reusable molecules which bring about chemical change while remaining unchanged themselves. They alter the rate of the reaction without altering the equilibrium. Remember, they only change the rate, but they do not alter the equilibrium. So you need to understand some important technical terms, like they are biocatalysts. That means they are present in living cells. In fact, they are manufactured by the cell and also regulated by the nucleus. But for their activities, they do not depend on the life of the cell. That means an enzyme can work outside the cell, and they are very specific in their reactions. And with the exception of a few catalytic RNA, which are also referred to as the ribozymes, all known enzymes are proteins. These enzymes, they differ from catalysts. Number one, how do they differ from other catalysts? They are produced in living cells. They do not depend on the life of the cell. That is, an enzyme can work outside the cell in a cell-free system. Number two, they are highly specialized proteins with extraordinary catalytic power. The enhancing the reaction rate by a factor of 10 to the power 5 to 10 to the power 17. If you see this reaction, water and CO2 will react together in the absence of enzyme to form carbonic acid, and approximately 200 molecules of carbonic acid are formed per hour. But when water and carbon dioxide react in the presence of enzyme carbonic anhydrase, which is one of the fastest enzymes, approximately 1 lakh molecule of carbonic acid are formed per second. Just imagine 1 lakh molecule per second. See the magnitude of rate enhancement. Then they are very specific in that they act either on a single or at the most structurally related substrates. The specificity may be for the substrates or for the type of bond or for specific isomer, maybe optical or geometric. Their specificity lies in the fact that they may act on specific type of substrate molecule, for example carbonic anhydrase, or it may be a group specificity on a group of structurally related compounds, for example lactate dehydrogenase, which catalyze interconversion of pyruvate and lactate, and a number of other structurally related compounds. Then the optical specificity. One of the most striking aspects of specificity of enzyme is that a particular enzyme will react with only one of the two optical isomers. For example, arginase acts only on L-arginine and not on its D isomer. Then geometrical specificity. Some enzymes exhibit specificity towards the cis and trans forms. For example, fumarase catalyze interconversion of fumaric and malic acid, but does not react with malic acid, which is the cis isomer of fumaric acid, or with D-malic acid. They also differ from other catalysts in that they function in aqueous solutions under mild conditions of temperature. In fact, they are thermolabile. and also under mild conditions of pH. They are very, very large sized colloidal molecules, and the size may range from 12,000 to 1 million kD. The integrity of native protein conformation is essential for their activity. And the last but not the least, they need to be replaced. Unlike other catalysts, yes, they undergo wear and tear. Unlike non-biological catalysts, they cannot last indefinitely in a reaction system, since they, being colloidal in nature, often become damaged or inactivated by the reactions they catalyze. Hence, they must be replaced constantly by further synthesis in the cell. Thank you.